Hello, my name is Dr. Rix, and today we will be talking about transcription. It is one of the key parts of the central dogma, and this class is all about learning about gene expression and genes and how to regulate gene expression. And so, in order for us to fully understand those very complex topics, we have to get to the crux of the central dogma and understanding the different parts of it. And that first part is going from DNA to RNA. And that process of creating RNA from a DNA template is called transcription. And that's what we will be discussing today. And so let's just kind of look at this structure and see what we remember from last week. And I'll just point out a few things. Hopefully, you recognize it as I go. One, that DNA is composed of nucleotides. And so this would be a nucleotide. These nucleotides are made up of a sugar, a base, and a phosphate group. Awesome. So that's one. The other thing that you should easily notice is that there's a pairing of purines and pyrimidines. And of these two, right, you have guanine here and cytosine here. Which of the two would be your purine? And along with me, you should say guanine would be my purine. I noticed two rings here, and this two ring structure is a dead giveaway for purines, and this one ring structure is a pyrimidine. So you get that, you see that? All right. Another thing that you should easily notice here is that DNA is anti-parallel. And so while one strand runs 5 prime to 3 prime, the other strand runs 3 prime to 5 prime, right? And so you see 5 prime to 3 prime on one end, 3 prime to 5 prime. And so this 5 prime end is, across from a 3 prime end over here. So anti-parallel is one other thing that you should notice. All right. This specific orientation is vital because it dictates how the RNA polymerase will read the DNA during transcription. So precision in DNA structure leads to precision in gene expression. So hopefully you caught all of those. A few more nuggets that you should easily catch is between the G's and the C's and the A and the T's, right? You see these dashes here. These dashes are what type of bond? Hydrogen bonds. And you should notice that there are 3 between guanine and cytosine, and there's 2 between adenine and thymine. Also, the sugar phosphate backbone held together by the phosphodiester bond. The structure of a nucleotide is essential, especially when distinguishing the differences between DNA and RNA. So here's a breakdown of the deoxyribonucleotide. And so first, I want to do it based on the carbon. So at the core is a pentose sugar. It's a deoxyribo sugar, and it's made up of 5 carbons. And these 5 carbons matter because there's a special, characteristic to each one of the carbons, except for one of them. But this first carbon is carbon number 1. And on carbon number 1, your base is always attached to carbon number 1. This is gold, which means that it'll be on the exam. You'll see this again. So remember this, right? But your base is always attached to carbon number 1. Carbon number 2 for me is that identifying carbon, meaning that from carbon number 2, I'll be able to tell if this is a deoxyribose sugar or a ribose sugar. And it's really about what's here. If it's an OH here, then I know that that's a ribose sugar. If it's just the hydrogen here, no oxygen, then that's a deoxyribose sugar. So carbon 1, the base is attached there. Carbon 2 is my identifying carbon. Carbon 3, if you notice there's a OH here, and whenever you see this, this is called a free OH. It's there where another nucleotide can bond. So carbon 3 has a hydroxyl group critical for the creation of the phosphodiester bond, and it's where a new nucleotide would attach. And so this is carbon number 3. That's where it's almost like Legos. It's like an open hole in the bottom of a Lego where I know I can add another Lego there. Carbon 4, I said, there's something special about all of them except for one. Well, the one is carbon 4. So there's nothing you need to know about carbon number four, but carbon number 5 is where the phosphate group will be attached. So the phosphate group is attached to carbon number 5. So carbon 1 is the base. Carbon 2 is identifying. Carbon 3 is where I would add another nucleotide, and carbon 5 is where the phosphate group would be attached. And a major thing that you need to remember here is that these nucleotides, so nucleotide is made up of a sugar, a base, and a phosphate group. However, and this is gold too, because you'll need this for the exam, if there's no phosphate group, then this is called a nucleoside. So you have a sugar, and you have a base, that's a nucleoside. But you have a phosphate group of sugar and a base, that's a nucleotide. Transcription is the first critical step in gene expression. Without it, the cell cannot produce the proteins, which includes what are proteins? They're enzymes, structural components essential for life. When transcription fails or enzymes are missing, we see an emergence of genetic disease. And you'll see this later on when we talk about certain inborn errors of metabolism or certain diseases that we'll discuss. You'll notice in most of those diseases, something happened at the genetic level, leading to something going wrong with the protein. And something being wrong with the protein will lead to genetic disease or other malfunctions. So let's look at the difference between RNA and DNA. So what are the key differences? Before we get into how RNA is made, we need to understand how it differs from DNA. The differences aren't just academic, they impact RNA structure, stability, and function. So RNA is typically single-stranded, it's more flexible, it can fold in different complex structures, while DNA is double-stranded, more stable, and primarily stores genetic information. First off, the sugar. All right, let's start off with the sugar. The sugar differences, and so they're both five carbon sugars, right? But I said that the second carbon is where we can see the difference between a ribose sugar and a deoxyribose sugar. And so if this is carbon number one, this is where the base would generally be attached. It's carbon number one. And then here is carbon number two. Here is an OH. That OH tells us it's a ribose sugar. And now I know that this sugar is involved in the creation of RNA. This is a deoxyribose sugar. I can tell by carbon number two having an H instead of an OH. And you can see no O, deoxy, right? Getting rid of the oxygen, deoxy. So the deoxyribose has just an H, and that H indicates to me it's a deoxyribose sugar, and the sugar is involved in the creation of DNA. Another thing about RNA that's different from DNA is that RNA has uracil instead of thymine. So RNA is chemically similar to DNA, but the major difference is in the bases. So instead of the thymine, which you'll often see ATGC, when you see the ATGC, that T indicates that it's DNA, right? However, when the T is replaced with the U, so AUGC, when you see uracil instead of thymine, it's a dead giveaway that it's RNA. So during transcription, when RNA polymerase reads an adenine on the DNA template, it pairs it with a uracil instead of thymine. RNA nucleotides follow the same purine to pyrimidine rule. So with purines are your adenine and guanine, and the purines are those two-ring structures, while the pyrimidines are still one-ring structure. You can see uracil has a one-ring structure as well. It is a pyrimidine replacing the other pyrimidine and thymine. This change, though small, affects RNA's structure and function, and especially its stability compared to DNA. Another thing you should notice here is between adenine and thymine are normally two hydrogen bonds, but the same thing applies for between adenine and uracil, it's two hydrogen bonds. Because RNA is single-stranded, it can fold into complex 3D structures using complementary base pairing. And so if you notice here, this A can pair with that U, this G can pair with that C. And because it can pair with multiple spaces on one single strand, so it can fold on itself. And that folding allows it to give you multiple different types of structures and also multiple types. And so there's tRNA and rRNA and even ribozymes. And those are RNAs with catalytic activity. And I'm going to show you some of those later as well. These structures arise from the conventional base pairing, A to G, G to C, and non-conventional interactions as well. This flexibility allows RNA to perform structural and enzymatic roles that DNA simply cannot. mRNA as a messenger RNA is a direct product of transcription. It carries genetic information from DNA to the ribosome for protein synthesis. rRNA, which is not involved in transcription, which we'll talk about today, but is involved in translation, forms the core structure for ribosomes and catalyzes peptide bond formation. tRNA, or transfer RNA, is involved in translation as well, not involved in transcription, and brings amino acids to the ribosome during translation. Of these, only mRNA is directly involved in transcription. It's the output we're producing when RNA polymerase is doing its job. Let's do a basic example here. Just transcribe that DNA sequence. You have 3 prime, you have A, C, T, G, A, C, T, G, A, C, T, G, right? 5 prime. Um, if that's the case, from there, go ahead and we're going to simply transcribe this. So I'll give you a second to transcribe it. Awesome. You should have come up with this, right? Where remember we said whenever RNA polymerase 2 encounters adenine and the DNA template, it's going to give you a uracil complement, and boom, there it is, right? It's still GC. There's no difference there. So whenever it sees cytosine, it gives you guanine, boom. And then but here's a kicker, and this is what I want you to be able to see. Whenever it sees thymine, it doesn't give you a uracil. Whenever it sees thymine, it attaches a adenine there. Okay, so that's one thing. The reason why we do this is so you can easily make that translation. So guanine cytosine, you see an A, add a U, you see a C, add a G, but you see a T, add an A. Thank you so much for following along. Have a phenomenal day. God bless.